Hello guys, in today's lesson we are going to talk about proteins. So proteins, why we have to learn about proteins? So what's the importance of that? Right. So proteins we need that in almost all our body functions inside our cells. Right? There are structural proteins, some are functional proteins. In functional proteins there are catalytic protein and storage proteins. There are various types of proteins. We will learn about all those functions in a bit later. Right, let's see what are the components or elemental composition of proteins. In carbohydrate, we had carbon, hydrogen, and oxygen. Here also we have all those three. In addition to that, we have nitrogen and sulfur. Right? Okay, and proteins are macromolecules. So what is macromolecule means? Macromolecules are the molecules which has high molecular weights. And they are also polymers. So when we say polymer, they are made up of monomers. Here the monomer of monomer of proteins is amino acid. Right. So let's see what's the basic structure of amino acid. So amino acid is the monomer or the basic structure of protein. Okay, now let's see what's the basic structure of amino acid. So we have a central carbon here, and in every organic molecules we have 4 bonds. So we have 4 bonds for the central carbon. In one terminal we have amino group. As the name suggests, amino acid, we have an amine group here, and as it says acid, that is carboxylic acid, we have with the other terminal, right? And we have a hydrogen here and an R group here. R group is also called as alkyl group or side chain. Okay, so in nature we have 20 different amino acids. In all those 20 different amino acids, this part is same, right? This is called the backbone. So this is gonna be same. Only the R group gonna change. So in the very simple amino acid, which is glycine, this R is replaced by hydrogen. In all the others we have other elements also, right? And as we have amino group here, that gives the alkaline property to this molecule, right? And here this carboxylic acid give acidic property for this molecule. As both alkaline and acidic property we have in this molecule, we call this molecule as amphoteric, or this molecule has amphoteric property, right? Okay, so in the 20 different amino acids, we said we have different R groups. So here we have a glimpse of that. You can see in this part we have electrically charged side chains. So you can see here we have positive, negative, negative. So we have charge groups. You don't want to know about the detailed structure of all this, but just you have to know there are different types of side chains. Okay, here we can see there are polar uncharged. They have polar groups like OH, NH, but they are not charged, right? And we have some special cases here like glycine. We said they have only single hydrogen. Here we have cysteine. We have sulfur here, SH. And we have a ring structure here, right? And these are with side chains with hydrophobic groups. That means they don't have any polar groups, they have just bulk of CH elements, right? Okay, we said proteins are polymers which are made up of monomers. Monomer is the amino acid, so amino acid has to join with one another with a bond, right? In carbohydrate we discussed, we had the monosaccharides binding with the glycosidic bond, right? Here we have peptide bond. Now let's see how this bonding occurs. Okay, let's say for example we have an amino acid here, right? We have NH2 COOH, right? We have H here, here we have R group. I'm just to differentiate, I put R1, and here we have another amino acid, let's say amine group, sorry, so we have hydrogen here. I'm going to differentiate using R2, and we have COOH, sorry for that, OH, right? Okay, I think it's clear, right? So what happens here, they undergo which reaction? In carbohydrates we discuss its condensation reaction. They release a water molecule, right? So OH from the carboxylic acid and H from the amine group binds together and they release a water molecule, right? Okay, so as they undergo condensation reaction, they release a water molecule and they form this bond between. Okay, although I have drawn long, it's just a single bond. Okay, so this is the peptide bond formed. Okay, now we have a molecule dipeptide which has two amino acids and a peptide bond, so we call it as dipeptide molecule, right? If it is a tripeptide, okay, let's using symbols. So this is a dipeptide, the circles are amino acids. If it is a tripeptide molecules, we have two peptide bonds and 3 amino acids. Although we use the word di here, this indicates the number of amino acids, not the peptide bond count. Okay, tripeptide, we have 3 amino acids, 2 peptide bonds. Okay, so and also we have, remember, we have a water molecule. So if they in exams, if they ask you to write draw the, if they will give you the amino acid structures and ask you to write the condensation reaction, remember to write the product as well, water molecule. Okay, so when there are numerous amino acids joined together by peptide bonds, they will form a polypeptide chain. We call it as a polypeptide chain, right? Like this. So this is a single polypeptide chain, right? So this polypeptide chain itself can be also called as a protein if they become functional. Okay, so sometimes in a protein there are more than one polypeptide chain. So a protein can be composed of a single polypeptide chain or more than one polypeptide chain. We will learn that in detail when talk about levels of protein. That's what we're going to discuss now. Okay, as we go through levels in our life, for example, before we become a job person, we have to go primary education, secondary, and some goes to tertiary. That means, it's higher education, and then they become a job person. Some they drop off from the secondary and they go for jobs, right? Like that, the proteins, before they become functional, they have to undergo some levels. So basically there are 4 levels they have. Let's discuss one by one, right? First one is the primary level. So in primary structure, we have just a linear chain of polypeptide, right? Just a linear chain, right? So we have to consider three things there. First one is number, order, then type, right? So we can remember it by using an acronym NOT, number, order, type. That means if the number of amino acids in a polypeptide chain varies, that means that's a different polypeptide. If the order, that is also different. Order is different, polypeptide chain is different. If the type is different, that's also different, right? Okay, let's see. For example, I'm using very simple symbols to indicate the amino acids. Let's say we have this, we have three types of amino acids here, right? And let's say we have, so here number is different, so it's a different polypeptide. Let's say we have here order is different, so that's a different polypeptide. And if the type is different, again different, right? So using one single word, we can call this number, order, type. You can use the word specific sequence of amino acids. Or we can also say unique sequence, right? Either word is correct, right? So that's primary structure. Okay, then secondary structure. In the secondary structure, it's like if you think about the amino acid chain or the polypeptide chain, you know, in the backbone we have, at one terminal we have the amine group, right? The other terminal we have the carboxylic acid group. In between there are lots of amino acids. Okay, let's say somewhere here we have another amine group, another carboxylic group, right? So in between these two carboxylic acid group, that is this oxygen and hydrogen, there will be a attraction, because you know this oxygen, they are polar, they attract the bond towards that, so they become delta negative, and this hydrogen, they become delta positive, because this nitrogen is more electronegative, so they pull the electron towards it, so this become delta positive, right? So this delta positive and delta negative, they will have an attraction force, right? So because of that attraction, what happens, these 2 points, right, here this point in this chain and this point, what happens, they tend to become closer. So because of that, now you can see there will be a bending, there will be a bending, right, like this. So now these delta positive and delta negative, they are close, right? Or sometimes they will form a curve, right, bending or curve. So because of this, in the secondary structure we can see bending and curling or curve. Okay, so we have two types of secondary structures. This is here I have given you the diagram. The first type, which is called alpha helix, right? So they have, the molecule is curved, right? So you can clearly see here what I said in about the bond formation, right? You can see CO and NH in between that, they form the, this is called the hydrogen bond. Okay, right, so this is the hydrogen bond. So the example for alpha helix is keratin. which is in your hair. And this is the next type, which is called beta plated sheet. Okay, here you can see there are bendings, and in between we have, here also we have hydrogen bonds, right? So it's kind of you have opened up a series of books, right, and kept, right? Although it's kind of the structure of the asbestos seeds as well, right? Okay, the example for beta plated sheet protein is spider's silk fiber. You know, spider's silk, yeah, that's a very strong. You have to be very careful here. This hydrogen bond is in the same chain, okay? Until in the primary structure also we have one polypeptide, single polypeptide chain. Here also we have a single polypeptide chain. So all the curves, curls, bends, everything occurs in a single chain. The next level is tertiary structure, right? So in the tertiary structure, what happens is the polypeptide chain, which has the secondary structure, they undergo further bending, and they will form a unit 3D, a compact structure, right? They undergo actually extensive bendings. Okay, so as they have to undergo extensive bending, they undergo several other bondings. Okay, in secondary structure we had the hydrogen bonds, which was in the backbone. Here we have 4 other bonds. Okay, we have to discuss one by one. First one we'll see, that's hydrogen bonds. Okay, so in the secondary structure we said the hydrogen bond is in between the backbone, that is CO and NH. Okay, but here the hydrogen bonds occur in R groups, right, in the side chains. If we discuss when we talked about the different amino acid side chains, we had polar groups. In some R groups we had polar groups. So those polar groups, they tend to form hydrogen bonds between amino acids. Okay, and second is disulfide bond, right? This is also called as sulfur bridge. Okay, so this sulfur bridge is caused by let's say in the polypeptide chain we have sulfur somewhere here and sulfur somewhere here. These 2 sulfurs, they can form this disulfur bond. Okay, so this is a covalent bond, so this is stronger than hydrogen bonds. In hydrogen bond is just an attraction. Here we have the actual bond. Okay, then the third type is ionic bonds. So when we discussed the different types of amino acids, some side chains have ions, plus or minus, or positive or negative ions. These positive ions, let's say we have in somewhere here plus, or here we have an O minus, like that. So this will form an ionic bond. Okay, because of that attraction, the bending or curving may happen. Okay, then we have the fourth one, van der Waals interaction, that is also called as hydrophobic interactions. Okay, so here that's also in side chains. If we think about the polypeptide chain, in the side chains we have non-polar groups, right? We said we have some non-polar groups in side chains. So these non-polar groups, right, they I'm just indicating with circles. So they have attractions between them. So what happens, they cluster together, right? So because of that, they form this curving or bending, right? That is called van der Waals or hydrophobic. This as we know, the hydrophobic means they repel water. Okay, that term you're familiar with. Most of the enzymes like enzymes which are biological catalysts, and myoglobin, that's also a protein which is in our muscles. Myo indicates muscle, right, that store oxygen in our body, and albumin. These are examples for this tertiary structure in proteins. Okay, so these proteins will have all the 4 bonds we discussed now. So as they have these types of bonds, now they will have a 3D structure, a compact structure. For example, let's say in alpha helix we had just curls, and in beta plated sheet we had just bends. Okay, like this. Here we will have all the types, bendings and curvings. So there will be a compact 3D structure, right? Okay, the 4th type is the quaternary structure. So quaternary structure is very similar to tertiary structure, but we have a big difference here. The difference is in tertiary structure, tertiary structure we had only one single polypeptide chain, right? But in quaternary we have more than one polypeptide chains, right? So that's the difference. So the quaternary structure, they also have all the 4 bonds we discussed: the hydrogen bond, disulfide bond, ionic bond, and the van der Waals interactions. So they also have this compact 3D structure. Okay, so the difference is the number of polypeptide chains. See an example for a protein which has quaternary structure. You can see here we have this molecule. This is the hemoglobin molecule we have in our blood circulatory system. So here you can see the 3D structure. If you look at this diagram, the B diagram, we can see there are 2 different colors, but we have 4 polypeptide chains. Okay, the top ones are alpha polypeptides, or polypeptides are called here subunits. Okay, alpha subunits, we have 2 alpha subunits, we have 2 beta subunits. So this is heme. Heme is the molecule which is in all the polypeptide chains. This binds with oxygen. We will learn about the structural detail in functioning animal, right? And collagen, which is in your bone, it's also an example for quaternary structure. Okay, so what is denaturation of proteins? So denature, that means they break down, or the loss of the 3D shape of the proteins. So this is the definition: loss of specific 3-dimensional shape due to the alteration of weak chemical bonds and interaction. So when we say weak chemical bonds, the bonds we discussed when we talk about the tertiary structure, we discussed about 4 types. Okay, so in those 4 types of bond, we said we have hydrogen bonds, ionic bonds, disulfur bonds, van der Waals. Okay, however, so those 4, disulfur bond is somewhat stronger compared to others. And remember, due to the denaturation process, the peptide bond is not affected, it's not broken down, it's not altered. Only these extra bonds are getting altered. Okay, and so this alteration or this denaturation occurs due to some factors. So you should know the factors. You don't want to know the mechanism, how they break down. So these are the agents that affect the denaturation. First one is high temperature and high energy radiation. Then strong acids, alkaline, and high concentration of salt. and heavy metals and the organic solvents and detergents. So you should know all these agents, and remember, you don't want to know the mechanism, how they affect. Okay, so finally we have to discuss the functions of the proteins. So you can see here we have given 3 columns type of protein, example, and the functions. First one is the catalytic protein. So you know catalytic means they catalyze the biochemical reactions. Example given is pepsin amylase. You will learn about lots of examples when you learn your functioning animal. Here pepsin, you know, it helps in the breakdown of proteins. Amylase, they also catalyze carbohydrates. So they have the common function as catalyze biochemical reaction. Okay. Then second type is the structural proteins. 2 examples are given. First one is the keratin. They prevent desiccation. Okay, that's in your skin. Okay, and in collagen, they are in your bones. They provide strength and support. Okay, and the third type is storage protein. We have given you 2 examples. First one is ovalbumin. So when we say ova, you should be able to understand that is they're talking about the egg cells. So that should be the storage protein in egg. Okay, then the casein, that is the storage protein in milk. The next type is transport proteins. Example, hemoglobin, you know, that transport oxygen and CO2 around your body, in your blood circulatory system. And serum albumin, they transport fatty acids. Hormones is the next type, where we here insulin and glucagon, these 2 are given. They regulate blood glucose level. So we will learn the detailed functions in when we learn homeostasis. Okay, and here I have to mention one important point. Not all the hormones are proteins. Okay, some hormones are proteins. There are non-protein hormones as well. So in part 1, if they give you the statement saying that all the hormones are proteins, that's wrong. Okay, then the next type is contractile or motor proteins. Okay, that example is actin and myosin. So you know myo, if the word myo comes, it's surely talking about muscles. That is the proteins in your muscles. So their function is the contraction of muscles. Then the last type is defensive proteins. The example is immunoglobulins. Their function is to eliminate foreign bodies. So when you talk or when we learn about immunology, we will learn the detailed function of these immunoglobulins. Okay, so basically these immunoglobulins, they can detect the foreign body. They act like signaling system inside our body. Okay, so yeah, that's all for protein. Bye until the next video. Thank you for watching.